Good afternoon, good morning, good evening, wherever you're zooming in from. I'm Barry Schweitzer, Senior Business Development and Licensing Manager for the Technology Transfer Office at Dartmouth College. And I'm excited and honored to be the moderator for today's program, Biotech-Academic Partnerships in Response to COVID-19. Before I introduce today's first speaker, I want to run through a few housekeeping items. First, please submit your questions through the Q&A button located at the bottom of your webinar window. We'll do our best to get to as many of those questions as we can. Also, please reserve the chat box for any technical challenges that you might experience during the webinar. So someone from the team will be monitoring that. This webinar is being recorded and will be made available at the end of today's session, actually probably early next week. You'll be receiving a survey at the conclusion of today's program. Please take a brief moment to provide us with some feedback that will help guide future programming. And finally, I just wanted to let you know that this session is eligible for CME credit through the Alumni Relations Office. So today we're going to hear from two outstanding researchers, one from the Geisel School of Medicine and one from the private sector, specifically a biotech company. In addition, I'll share a bit about my role here at Dartmouth and an exciting new initiative that we're calling the Dartmouth Innovations Accelerator for Cancer. First, it is my pleasure to introduce our first speaker, Dale Chappell. Dale earned his medical degree from Dartmouth and his business degree from Harvard. In 2002, he founded the investment management company Black Horse Capital, where he serves as director and manager. In addition, Dale serves as Chief Scientific Officer for Humanigen, a biopharmaceutical company that develops next-generation cell and gene therapies for the treatment of cancers. Dale, please begin your presentation.
Great. Thank you, Barry. And good afternoon, everyone. Thank you for dialing in to today's webinar. My name is Dale Chappell. I'm the Chief Scientific Officer of Humanigen. I'm going to walk you through a little bit of my background, and then we can get into cytokine storm. What is that, how it's related to COVID-19, and why we decided to pivot into COVID-19 as a therapeutic area. We could jump to, yeah, there we go, perfect. Yeah, so let's jump to the background slide, and I'll walk through a little bit of the background, and then we'll get into the cytokine storm aspects. Yeah, perfect, thank you, Bridget. So I'm a proud alumnus of Dartmouth Medical School, class of '99, went on and got a business degree at Harvard. After that, did a stint as a Howard Hughes Fellow at the National Cancer Institute, where I was in the laboratory of Steve Rosenberg. He's considered one of the fathers of immuno-oncology. In his lab, I was studying tumor immunology, and specifically looking at this molecule, this signaling protein called GM-CSF. And that's going to be important as we get into cytokine storm, because it turns out that GM-CSF is an important initiator of that storm. So after my stint at the National Cancer Institute, I joined an investment fund in New York City. And then in 2002, started my own investment firm, Black Horse Capital. At Black Horse Capital, we take very concentrated positions in only a very small few companies at any one time. We get very involved with the companies, and in this particular situation with Humanigen, I've joined now as a Chief Scientific Officer and part of the management team. At Humanigen, we've been developing a therapeutic drug called Lenzilimab. We've referred to it as LENS for short, so it's not quite such a mouthful. We've been positioning this drug as a therapy for cytokine storm over the last 3 years. So cytokine storm is an overwhelming immune reaction where your body essentially secretes too many cytokines. This can lead to hypertension, it can lead to organ dysfunction, and it occurs in other sort of fairly dramatic settings. Two fairly new cancer therapies, one called CAR-T, it has a side effect of cytokine storm. We also see this in allotransplants, where some patients can develop what's called graft versus host disease, and that's a form of cytokine storm as well. So we've been developing LENS to treat these toxicities in these two different cancer therapies for several years. Then in late February, data out of China started to suggest that patients with COVID-19 were suffering from cytokine storm as well. So it's clearly there's some pathology that's directly caused by the virus, but what appears to be happening is that the immune system's reaction to the virus, or in some instances, the overreaction of the immune system, leads to some form of a cytokine storm, and that can cause respiratory distress, respiratory failure, can lead to patients then becoming ventilated. So let's talk about sort of the pathway of cytokine storm, and yeah, we're on the right slide. So this is a schematic that we're looking at of the immune response in COVID-19 that leads to cytokine storm. We're showing in orange a particular type of immune cell called a T cell. When these T cells see a virally infected cell, they start to secrete a signaling protein called GM-CSF. Now, GM-CSF, that's a, it's a long name, stands for granulocyte macrophage colonase stimulating factor. You don't need to know that, but just know it's a signaling protein that activates another type of immune cell, or these myeloid cells that we're showing here in red. Now, myeloid cells are very important in cytokine storm because they secrete a whole host of downstream cytokines that really are responsible for the pathophysiology of the storm. So you'll see in this diagram, IL-1, IL-6, IL-8, those are all responsible for some of the effects that you see on the right: hypotension, the respiratory failure, the multi-organ failure. But the key to initiating this process is GM-CSF. And some of the earliest work that was done to identify GM-CSF as the initiator of this process was done in the lab at Dartmouth under Charles Sentman. So he published some work showing that GM-CSF was actually very important in initiating this process. And you will see then what we're trying to do with LENS. LENS is depicted here as a black antibody. We're trying to block or neutralize GM-CSF to prevent this storm from ever really starting. So let's jump to the next slide. So since we were already thinking about cytokine storm and patients with COVID-19 were experiencing cytokine storm, we decided to pivot into that therapeutic area. And what we're seeing here is top-line data. This is the first 12 patients who were treated under single-use emergency INDs, often referred to as compassionate use. These patients were treated at the Mayo Clinic. And what we're looking at is the time to improvement. So this is a recognized hospital scale. Patients have to improve by 2 points on this scale to be considered improved. And what we're looking at here, and again, it's a small subset of patients, only 12 in our cohort. It's not randomized. We're doing cross-trial comparison. So with all those caveats in place, it looks like we're seeing something fairly interesting with linzilimab. Our patients are going home in a median of 5 days. They're showing recovery in 5 days, and that compares to remdesivir, which is a direct-acting antiviral, where you're seeing improvement in about 11 days. And so remdesivir's received FDA emergency use authorization for that 11-day time to improvement. And in our small cohort, we're seeing time to improvement about 5 days. So this is obviously very exciting. It's very early. We can't draw too many conclusions from this data, but this is exactly what you'd be hoping to see at this early stage of development. So let's jump to the next slide. So we've now planned it's a multi-center, 238 patient phase 3 clinical trial. So it's randomized, double-blind, placebo-controlled. We're looking to a number of different clinical sites. Some of the academic sites that we've been able to sign on to this trial include Dartmouth-Hitchcock. We're very happy and thrilled to have them as part of this clinical trial. We also have Mayo and Stanford, who are now active in enrolling patients. And I have to say, it was interesting. I got a press release this morning from Dartmouth-Hitchcock saying that the site initiation went from start to finish in 8 days. That has to be some sort of record. So certainly thrilled with the speed at which Dartmouth-Hitchcock is moving, and we're very lucky to have them as a center. So I'll hand it back to you, Barry, and look forward to your questions during the Q&A session.
Thanks so much, Dale. That was really exciting, and really appreciate you taking the time to share that with us. We wish the best of luck in that trial, all those trials. I'd like to now introduce David Leib, Professor and Chair of the Department of Microbiology and Immunology at the Geisel School of Medicine at Dartmouth. Dr. Leib studies the pathogenesis and biology of herpes simplex virus, or HSV, with an emphasis on the interaction between virus and host. His lab has generated many discoveries and innovations, including recombinant viruses that are effective as vaccines for the prevention of recurrent HSV infections. David?
Thank you, Barry, and thank you for the opportunity to talk today. and coming to you from beautiful sunny Etna, New Hampshire, from my home. I've had quite a long journey professionally. I grew up in England, as some of you may still be able to tell. I went to the University of Birmingham, that's Birmingham, England, not Alabama, and then did my PhD at the University of Liverpool before transitioning to a postdoctoral fellowship at Harvard University. And then in 1990, I joined the faculty at Washington University in St. Louis, where I spent 20 glorious years working on herpes virus infections, actually of the eye. In 2009, I was recruited to Dartmouth, and I joined the Department of Microbiology and Immunology, and I became the chair of microbiology and immunology in 2018. Next slide, please. So my day job, in addition to being chair of a very busy basic science department, especially busy, as I'm sure you can imagine right now in these days of COVID-19, as Barry pointed out, I'm a virologist by training. My specialist area is herpes simplex virus. In particular, right now, we're interested in neonatal infections and how we can use therapeutic antibodies and vaccines to prevent lifelong disabilities that result from these neonatal infections. We're also interested in prevention of neurodegeneration, since a recent association has been drawn between HSV, herpes simplex virus infections, and neurodegenerative diseases. On the next slide, it will describe, and I will talk about how we pivoted to working on COVID-19 during the early days of the COVID pandemic. And so what we learned early on was that there was a major bottleneck in terms of testing for COVID-19. And this bottleneck actually, to some extent, still exists. So the CDC test that was originally developed early in the year was slow, relatively insensitive, and relatively expensive too. And it had a very high false positive and false negative rate. But the biggest obstacle was that it relied on the so-called extraction reagents that were required in order to get the viral RNA out of the sample and then into the test. So these extraction reagents became limited, which became an additional rate limiting step for the testing. So we were approached by a biotechnology company in California known as Atila Biosystems, and also by Covelocity, more on them in a couple of slides. They had developed a test that was very quick, it was cheap, it was sensitive, low false positive, low false negative, and it was very amenable to low resource environments. Again, more on that later. So they had developed this test, but as of that time, it was not FDA approved and had never been trialed on actual patient samples. It had only been trialed in a laboratory setting. Next slide, please. So at this point, we swung into action. We felt a moral imperative really to get involved to try to solve this problem of the testing supply line. Audra Sharon, pictured here with me in the lab, is a senior research associate who's really a kung fu molecular biologist. Audra and I also happen to be married. And so this allowed us to work together without the need for social distancing. And this, I think, was important for moving the work forward. The other part, I think, that was really important here was that Dartmouth opened up very quickly to give us the regulatory approvals that we needed, because actually at this time our lab was closed due to the COVID-19 pandemic. We were also able quickly to establish a collaboration with Joel Lefferts at the Dartmouth-Hitchcock Technology Lab in the Williamson Translational Research Building that was right next door to the building in which we worked. So Dartmouth's nimble and cross-disciplinary environment really enabled us to very quickly get on track to do this testing. So what we were able to do once we got a hold of patient specimens was to compare the new Atila test head to head with the older, slower CDC test. We obtained patient samples that were coded and blinded, so we didn't know which sample was positive and which was negative. We ran these samples through the Atila test. And then once we cracked the code, we found complete concordance between the new test and the old test, only for a lot less money and with a lot higher speed. And because of all of these factors together, within 3 weeks, we were able to bring the data and bring the test to emergency use authorization with the FDA. And this test is now running live in the Dartmouth-Hitchcock Clinic. I mentioned earlier that we've been working with a company, Covelocity. Covelocity is a company which has a philosophy, it's a non-profit, which is committed to bringing infectious disease diagnostics to the world for use in low resource settings. One of the things that you might notice from looking at the Johns Hopkins map of incidence of COVID-19 throughout the world is the low incidence of COVID-19 in Africa, parts of South America, and parts of Asia. We don't believe that this is due to actual incidence, but rather due to a lack of testing in these underdeveloped places. So Covelocity is committed to the idea of bringing these low-cost tests, PCR machines, and more importantly, training for people on the ground to be able to run these tests in countries such as El Salvador, Haiti, Nigeria, Ghana, Zimbabwe, and Paraguay. And actually the list now is beginning to grow. So with that, I'm finished with my presentation and happy to take questions later, but back over to you, Barry.
David, that's really great. I think we're seeing a theme here of how Dartmouth responds with speed and flexibility to changing circumstances. I'd now like to take a few minutes to share how biomedical research at Dartmouth is translated into the marketplace, which is a key step in scaling up that innovation for bringing it to the patient's bedside. So first of all, at least the most important thing to me is that I'm a proud member of the Dartmouth class of 1982. After Dartmouth, I went to Yale to receive my PhD in pharmacology and toxicology. I then spent some years in academia as a faculty member in chemistry, then came back to New Haven to help start up several biotech companies, and then stayed there in both startups and in global biotech companies for about 15 years before joining a venture capital firm about 5 years ago. And then coming full circle about a year ago back to Hanover to join the technology transfer office as senior business development and licensing manager. I'm part of a really great team that includes Kim Rosenfield, our director of tech transfer, Jamie Coughlin, the director of the Magnuson Center of Entrepreneurship, and our associate provost for entrepreneurship and tech transfer, Eric Fossum. Eric is the inventor of the CMOS chip and a real role model for entrepreneurship at Dartmouth and other places as well. So I just think I have a really fantastic job. Not only do I have a great team, I live in a wonderful and beautiful Upper Valley of, I live in Vermont, but get to commute to Hanover, or at least I did until working remotely. And I'm responsible for the commercialization of all the life science research that goes on at Dartmouth College and Geisel School of Medicine, which means I get to see all the cool new science that comes from our faculty, our staff, and our students. I work with the inventors to see if they can be if these technologies can be patented, and then I try to get these technologies out into the world where hopefully they can do some good. And sometimes that occurs by licensing to existing companies, and sometimes that it means working with investors and the founders to start up new companies, which is probably the most fun of all. As you can see on the table on the right, we've been pretty successful at doing this. Just in 2019 alone, we filed over 100 patents. We licensed 16 different technologies to companies. And very importantly, we worked with faculty to start up 6 new companies. And I'm happy to say that this year we're on track to meet or beat those numbers, even in the time of COVID-19. And speaking of COVID-19, just wanted to let you know that we're a signatory to the COVID-19 Technology Access Framework, which means that we're committed to bringing COVID-19 related Dartmouth technologies to the marketplace through easy and fast licensing strategies to provide global access to these technologies as quickly as possible. And I can't go into the details of that, of some of this, but I can tell you that we're already working on agreements for some of these technologies, some of which are in the clinic or will soon be in the clinic. And as someone who loves this small college, I take a lot of pride in that, and also no small measure of comfort. So if you can go to the next slide, please. So I would just say that innovation at Dartmouth, technology transfer from Dartmouth to the marketplace, and Dartmouth entrepreneurship is alive and well today. But could we do better? Of course. So that's why I'm excited to tell you about a new $15 million initiative that's part of the Call to Lead capital campaign to create the Dartmouth Innovations Accelerator for Cancer. This is in partnership with the Norris Cotton Cancer Center and the Magnuson Center for Entrepreneurship. Why these places? Well, I'm sure many of you have heard about the fantastic entrepreneurship programming that gets carried out by the Magnuson Center for students, alumni, and faculty and staff. But you might not be aware that the Cancer Center is the epicenter for entrepreneurship at Dartmouth School of Medicine. And that's shown by that we're ranked number 6 in the nation for small business innovation research grants, which is truly remarkable. Another, you might also say, well, why now? Well, frankly, this is a great investment by the college. Dartmouth College has an impressive track record for bringing new therapies to patients. And so, in fact, as a result of that, the revenues from licensing these technologies makes up the majority of what comes into Dartmouth. I believe that through bringing funding to the most promising research projects coming out of the Cancer Center, mixing that with training and mentorship in bioentrepreneurship, we can unleash the full potential of biomedical research at Dartmouth and bring those innovations and breakthroughs to patients suffering from cancer as well as other diseases, including COVID-19. So with that, I will move to the second part of our session, which is the Q&A. I'd like to start off with a question to Dale. Dale, how do you go from being a physician scientist to becoming an entrepreneur and chief scientific officer of a biotech company?
Yeah, Barry. Well, I wish I could tell you it was some grand plan and it was all planned out, and that's why I did what I did. But the reality is it was pretty much a long journey of me just following my interests and probably being brutally honest about whether I was happy with the path that I was on. So early on as an undergrad, I knew I loved science, but not all science, particularly biomedical science. And that's really what drove me to medical school at Dartmouth. While at Dartmouth Medical School, I absolutely loved the first 2 years. So those are the basic science years, but was less keen about the last 2 years, which are the clinical science years. And I have to say that was probably the exact opposite reaction of most of my classmates. They absolutely loved the clinical medicine and kind of got through the basic science. So I knew at that point I needed to do something that was really more basic science driven. And so that was kind of the impetus that drove me then to the NCI to start going down more of a science path. Certainly with the MBA degree, it lends itself to doing things around industry. And so, I wanted to do something that was a little more flexible than just academic science. And, I have to say, being involved in a small biotech company and that entrepreneurial aspect, I find absolutely fascinating. I love what I'm doing. You can be reading science papers in the morning, having discussions with regulatories and key opinion leaders, physicians in the afternoon, and then talking to potential investors in the evening. So it really allows you to use this huge skill set. And I think that's true of everybody who's in a small company, is that, you know, you can't be one dimensional. You have to be multifaceted. And so for people who want to have that flexibility, I think, you know, being in a small company, being an entrepreneur sort of lends itself to that. So I guess at the end of the day, it was really following my interests.
Fantastic. David, question for you. How does a virologist working on herpes jump into COVID-19?
Thanks, Barry, for that one. A number of things, I think, drove me to do that. I mean, first and foremost, a lot of the techniques in virology are common and fundamental to all viruses. And so that allowed us to easily, technically, make the switch. But there were a few other motivating factors. The first one, which I alluded to during my talk, which is that this extraordinary lack of preparedness, frankly, for testing large numbers of people made me suddenly very motivated to do something. My lab was shuttered. I was at home mostly shuffling emails around and needed really something to get my teeth into. And so that was a very strong motivation. The other motivation that came a couple of weeks into us initiating the work, so it wasn't an initial impetus, but certainly drove us forward, was that both my sister and my first cousin living in London went down with COVID. And if there's anything that can motivate you as a scientist, it's having a close loved one or family member coming down with a particular disease. So this was certainly two very motivating things. I think the other thing which drives me as a virologist is that we don't know much about this virus yet. We're still really at the infancy, really, of our knowledge. And as a virologist, this one's a little bit different. It's defied all the predictions and all the projections. And so I couldn't resist the intellectual challenge of getting involved.
Great. Here's a question that I'll ask both of you to take a shot at this. So start with you, David. This comes from the audience. Governments, healthcare systems, and biotech are fast-tracking research and clinical trials for COVID. Do you think this pace will continue?
That's a great question. I'm going to kick that one over to Dale first. I'm happy to come back on it, but that's more his area than mine.
Sure. I think given the immediate need, as long as the pandemic continues sort of on its current pace, given that immediate need, I think there'll continue to be an impetus to move quickly across academia, regulatory bodies, and then industry as well. If we start to see some fall off and maybe there's not as much need because people are able to get back out and start doing their normal daily activities, I am concerned that some of that, I guess, immediacy will start to fall away. But I think as long as we are where we are in the current pandemic, I see this moving ahead as is, which I think is a good thing. We're making a lot of progress very, very quickly.
So David, I won't let you off the hook. I'll just tweak the question a little bit in terms of, we have seen research funding, being, moved, quickly, fast track research grants. Do you see that as, for COVID, do you see that continuing?
So I think it is going to continue for the foreseeable future. I think there's a little bit of a danger, however. Clearly, in the middle of a pandemic, you can't overinvest in trying to solve that problem. But it doesn't mean that the other biomedical problems go away. And so one concern that I have is that with the extraordinary amount of attention being focused on COVID, what happens to cancer? What happens to other infectious diseases that are still out there that might well still be there, big if not bigger, when COVID is gone? And so that's a concern that I have of sort of, if you will, an overinvestment and an overemphasis in this area.
If we look at that a little bit differently, have we learned anything from this in terms of how to speed innovation, from COVID to other diseases like cancer?
Yeah, I think that's a really good thing to think about, because this has been a game changer in terms of the way that, for example, the FDA has moved forward with its emergency use approvals. I mean, we got this thing approved in, less than 3 weeks. Who ever heard of that? And I'm just wondering, if more people are going to be saying, well, if we did it during COVID, why can't we do it now? Why can't we do it, at another time? And so while I agree with Dale that, the immediacy of COVID has driven this, I'm wondering if we might not see some more permanent and actually positive changes on the regulatory front. Dale, what do you think?
Yeah, I hope so, David, because, there's conditions, as you said, like cancer. So one of our trials is for relapsed refractory lymphoma patients. And as you said, those patients are not going away. They still have that disease. And, I have to say, getting that trial up and running and now enrolling, that process took about a year. And we were able to do the same thing in COVID-19, probably even at a larger scale, more patients, real phase 3, more centers, in a matter of weeks. So it does tell you that it is possible, particularly with drugs that have already proven safety, right? So I think, and hopefully there's a lesson there that we can take into the future, even when this pandemic goes away, that for some of these diseases where the medical needs dire, where you have drugs that have already proven safety, you should be able to move at a much quicker pace.
So Dale, I was struck, on that note, I was struck by what you said in terms of 8, and what I read, as you said recently, regarding 8 days to get ready at Dartmouth-Hitchcock for the trial of your drug. Can you explain any thoughts about how that happened? What happened? How they make that happen?
I'm not sure. That's actually a great question. Maybe there's one of our audience members who was involved with that. But we've seen other sites, they have COVID-19 IRB specific boards. So those are institutional review boards that review clinical trial for ethical safety, type considerations. So they have boards that are sort of pre-convened and looking only at COVID-19. So that speeds up the process. Some of the contracting that we're doing, there's, industry contracts that as long as the site or the company don't make any changes, that you just sign and go. We've been trying to do that with all of our sites so you don't get into this legal morass of one side making changes and the other side sending it back to legal and back and forth. So again, there are probably some lessons here in terms of how quickly we can get sites up and activated, but Dartmouth, that's sort of light speed. I would love to know how they were able to do that in 8 days.
And so one other question that I would like to ask in that regard, do you think that really, I guess maybe for hope, God forbid we have another pandemic, but do we, can we speed up even further from what we're experiencing now, both on the therapeutic side, Dale, and on the diagnostic side, David?
So I think on the diagnostic side, we've learned a lot from this pandemic. And I think that one of the biggest things, of course, we've learned is that testing and incidence are not linked if testing is limited. Mm-hmm. And so the epidemiology of this virus is such that it was able to spread very quickly through untested communities. And it should be possible with today's technology to pivot incredibly quickly to develop and distribute these new diagnostic tests. And I think we've learned a lot about how that can be accomplished during this pandemic. I mean, necessity is the mother of invention, it's said. We've had a huge necessity and there's been a lot of invention. And I think that this will hopefully carry forward and we will learn a lot of lessons.
Yeah, Barry, I guess from the therapeutics side, I would say hopefully something that we're learning in terms of the number of clinical trials that are being done. If you look at it, there's thousands of trials going on in COVID-19. And if you start to parse those back, you look for trials that are randomized, blinded, multi-center. It's a small fraction of those trials. So most trials are really not set up to give you an informed answer on efficacy and safety because they're not randomized. And some are not even powered appropriately. So there's not enough patients. Many of those trials that are listed on clinicaltrials.gov are 50 patients or less. So it's impossible to get a real answer from those clinical trials. So hopefully some of the learning that would come out of this would be a little more, I guess, collaboration across the regulatory, academic, and then industry, that we're running trials that are going to give us a real answer. So we know, a quick no in a pandemic is better than an uncertain answer when you run a clinical trial. So hopefully that's one of the lessons, is let's power the trials. Let's do the right trials. Let's get randomized controlled trials so we actually get real answers and can move forward one way or another.
So David spoke about the need for low cost testing in third world countries and other more sparsely populated countries. Dale, do you think, what about therapeutics to those companies? How do you foresee Lens potentially making it there, or what about, if not, what about other treatments?
Yeah, sure. So with Lens, Lens is a biologic. So it's a monoclonal antibody. It's made in a human mammalian cell culture in these very large bioreactors. This is an expensive process. So that would require something like a Gates Foundation to be willing to step up and do large scale manufacturing to be able to supply that at some reasonable price for, at a low cost for third world countries. The other alternative would be is once you've proven that cytokine storm is a concern, and actually if you inhibit that pathway, you can blunt the serious side effects, or you can blunt the severity of the disease and prevent patients from progressing to respiratory failure. There are orally available drugs that are less specific, like some of the JAK inhibitors. So I think the FDA is looking at that. They're looking at a combination of a JAK inhibitor with remdesivir. Those are oral drugs. They're small molecule drugs, so they're easy to manufacture. They're cheap. That may end up being more of a solution for a third world type situation where you could supply those drugs cheaply.
Okay. So I just want to, there's a question that came from the audience. It's sort of to me, so I'll just want to make sure we get to that. But then we'll get back to some of the other questions. One question was how, if you're, let me just see if I can pull it up here. It's wonderful to see a partnership between academia and industry, fostering innovation at Dartmouth in alignment with patient care and investor return. How can those of us who love biotech entrepreneurship and the big green get involved? Great question. Certainly it's one near and dear to my heart. Basically, reach out to me. We're definitely looking to, really, with the accelerator, we're going to be looking for alums to help mentor entrepreneurial teams. We're going to be looking for entrepreneurs and residents. And now, with Zoom, we don't really actually need to be in the same place. So I think there's lots of opportunities for people involved, for alums involved in biotech, to get to interact very closely with my office and with the Office of Entrepreneurship and Tech Transfer, and just contact Alumni Relations if you want, or contact me directly. Love to talk to you. Okay, getting back to some of the questions. Let's see if I can get that up here. Wait a second. Oh, there we go. So, David, has at this point the Atila test hasn't been deployed to Africa yet, has it?
Not yet.
Okay.
We are about to deploy it, however, to Paraguay.
Okay.
I had a Zoom, what else, with the health minister of Paraguay. It was quite an experience, in which he was asking me about how the Atila test stacked up in terms of its performance, because we obviously have a lot of experience with that. So we're in the process of working with agencies on the ground, albeit either non-governmental or governmental agencies that are asking us for our advice on how best to use this test.
Okay. David, do you know whether anybody here at Dartmouth in our faculty have any connected at all with the Bill and Melinda Gates Foundation on any projects?
So Bill and Melinda Gates, yes, there are a couple of faculty members in infectious diseases working in the tuberculosis field who have connections to the Gates Foundation. And it's something which I know many of us in the infectious disease community have talked about reaching out more to them. It's interesting what Bill Gates, I think it was, who has said about the current pandemic, which is that, you know, if we were as prepared for this pandemic as we were for nuclear war, we'd be in a different position.
Right. Dale, you mentioned that you're getting ready to launch phase 3 trial, and then then there'll be again further time and so forth for all the regulatory. What do you think is best case for assuming trials go well for Lens to actually reach reach the marketplace?
Sure. So the trial should have an interim analysis. So that's when about half the patients have been enrolled. The FDA's asked us to stop enrollment and take a look at the trial, both for efficacy to see if it's working and for safety to make sure it's not doing any harm. So that should happen probably sometime in late July. And then the trial will most likely continue. It will be fully enrolled by the late summer. So probably by early September, we'll have a good read on whether this is working with the full data set. So, and then it will depend on the FDA. We're certainly prepared to go down the biological licensing application process, but there's the roadmap that they laid out with remdesivir, where they gave it an emergency use authorization ahead of a formal application for approval. So we think that's a possibility. If we're talking about best case scenarios, that would be one of the best case scenarios, is you get an emergency use authorization even before you're applying for full approval.
Okay. David, we've talked a little again about speed, and it looks like he's on. Can you unmute David? Oh, there he goes. David, can you hear me?
Step off camera for just a moment.
Oh, there you go. All right, you're back.
I'm back.
So again, we've talked about speed on the therapeutic side, speed on Dartmouth side. What do you think? How can we speed up diagnostic tests at Dartmouth or else?
So I think technology is a big one. We, with Atila, have been helping to optimize a new PCR-based, polymerase chain reaction-based technology that has cut the amount of time needed for PCR-based diagnostics basically by 20%. So we're down 80% from where we were. So a test that would normally take about 4 hours is now taking about 40 to 50 minutes. There are a large number of companies now which are developing these quickie tests that can be done in 10 to 15 minutes. But the reliability of these tests in terms of false positives and false negatives still seems to be a little bit of a problem. But I think the hope is that with increased technology, we will have much faster diagnostics, and that's really going to help, particularly when you think of hospital admissions and so forth. Return to work, being able to quickly screen people is going to be very helpful.
Okay, a question came in regarding, this is one that's certainly, I mentioned that that Dartmouth has signed on to the open framework for COVID-19 from a technology transfer point of view, where we will, we commit to give essentially limited time royalty-free licenses to companies who want to license Dartmouth COVID-related technologies. But the question came in is, given the significant public investment in development of COVID-19 therapeutics and diagnostics, Should there be a limit on the costs and profits from the applications of these new developments? This would, or maybe should, limit the investment interest of most venture groups. Is this a potential limitation to successful commercialization? Thoughts about that, Dale?
Yeah, I think it's a great question. In the instance where BARDA, for example, were to provide significant amounts of funding, I think that could be built into the contract with the company. Certainly from an investor standpoint, if you're putting up a small percentage of equity and then you're getting non-dilutive financing from BARDA, the return that you need then is much, much smaller than what it otherwise would be if you had to fund the entire amount. And some of these drugs, including LENS, they're expensive to manufacture. It could be hundreds of millions of dollars just from manufacturing costs to get it ready in a commercial supply to supply the US and global market. So, if BARDA's funding that, there's a contract between you and BARDA. They could actually negotiate what the price would be ahead of time. And I certainly think that's doable and would provide a return to your investors because, again, they write much smaller checks. So they don't need to have such a huge return on the backside of that. So I think that would be a decent model. I don't necessarily think that that's what's happening, but it's something that could be implemented.
David, any thoughts on your side on the diagnostic side, which is even a kind of in some ways a tougher on the business side because just the profits really aren't the same.
Right. So I think that particularly with what we've been working on, it's a special situation of high acute need. We have no idea how long that need is going to last. There's also, of course, the fact that regardless of profit, this is something which the world recognizes is absolutely a need for us to return to any kind of normal life, commercial or otherwise. So I think in this particular case with COVID, it's kind of a special situation than perhaps when you're looking at a highly specialized cancer therapy that might be designed to treat relatively few individuals compared to what we're facing with this pandemic.
Okay. Again, I'll stay with you for a second, David. So speed, you talked about speed and how that was good in terms of, for Atila and other companies to get the emergency use authorization. And again, I don't know exactly, I haven't seen a whole lot of scientific reports on this, but there have been at least some claims in the media that some of these tests are not as high as they should be in terms of accuracy. How do you balance the the need to get this stuff out there with accuracy?
This is one of the key questions that I'm hearing time and time again in the scientific community. First of all, as it pertains to FDA approvals, these emergency use protocols that are coming out really by the dozen every day because of this very, very high and acute need. It's also something that's being debated by scientists who are surveying the scientific literature right now because a lot of scientific journals are accelerating publication of anything COVID related. So the question is whether corners are being cut in terms of peer review of this information. So is everything that's coming out during this pandemic accurate? And it's a question of whether, the need for the information and the need for the diagnostic test to some extent trumps a need for complete accuracy. This is a very, very difficult question to wrestle with. I would posit that when it comes to false negatives and false positives with testing, the bar needs to be really high. Because certainly if you've got false negatives during a pandemic who are going out in the community, they've tested negative, but they're actually COVID positive, the damage that can be done is enormous. So I think we have to be very, very careful.
I mean, I think certainly both on the therapeutic and the diagnostic side, we're trying to interpret these results when we don't even fully understand the biology of the virus or even both its effects, how it works, what its structure functions are. So it's a little bit like searching, a little bit about reaching around in the dark, making it easy.
Absolutely. Which is why I think that, I think that some people have thought that certain measures are draconian or overreacting. And I think that really that's the only rational thing to do when we have so little good information about this virus. But things are changing.
Dale, on the therapeutic side, do you see increased tolerance for, say, maybe a lower therapeutic index for some of these drugs, or for treating these patients, or how do you in terms of other diseases?
Yeah, and I think it depends on the severity of the patient. So we've seen, for example, some of the immunomodulators, and I'll talk about the IL-6 inhibitors specifically. There are several of those that are already on the market, and they have black box warnings. That means on their prescribing label, they have kind of a box that's highlighted in bold for infection risk, and particularly pulmonary infection risk. So typically, somebody with a viral pneumonia, you certainly wouldn't be thinking about giving an IL-6 inhibitor. So I think what the FDA has done in those situations is to relegate them to patients who are critical. So if the patient's already mechanically ventilated, obviously the prognosis is not great in that situation. So you may be willing to go ahead and try something that otherwise would be contraindicated in the setting of a viral infection. But given what we know so little in this setting, the fact that it has the potential to give some benefit, they're relegating it to patients where the risk benefit, I guess, is more in line with the black box warning. But in normal times, I don't think you would see that. I think there would be sort of a prohibition of using those agents in the setting of any type of viral infection.
And so staying with you, Dale, in terms of, I think it's kind of a related question in a sense, will the COVID clinical trials of LENS ultimately benefit its development as a cancer drug?
Possibly. I mean, clearly there's a read-through there. So even some of the data that we've already received, when you look at other biomarkers, so, we're neutralizing GM-CSF, you can see whether, are you downregulating these other cytokines that are downstream? In other words, the mechanism as we drew it up on that little diagram, does it appear that that's how it's working in humans? So there will be those types of read-throughs in terms of, if we're impacting cytokine storm in COVID-19, all of a sudden the probability of success will go up in our CAR T trial where we're trying to prevent cytokine storm in that setting as well. So, I think absolutely. And even on the safety side, so on the dosing, what we're doing in COVID is very similar dosing strategy to what we'll be doing in our CAR T clinical trials. So there's a safety read-through as well. So if that dose is safe in COVID-19, it's more likely than not that it's going to be safe in the setting of CAR T.
Cool. Back to you, David. Now, we're starting to hear a little bit of good news, very early, maybe good news on the vaccine side of things. Do you see the testing, I haven't really heard of any, obviously they're looking at antibodies and, in terms of antibody tests and things like that, but I haven't really heard much of tests that would be specifically matched with or utilized to look at the efficacy of vaccines. Do you think that's an area that's, that is, is that being overlooked, or is there enough overlap between what's going on now and, and the development of those vaccines?
I think there's a lot of work being done in this area. It just has yet to be written up and publicized. I think it's just too early. Work done actually right here at Dartmouth. Margie Ackerman, Ji-Won Lee, Peter Wright, and others. They're working on exactly this issue. And I'm sure that once these experimental vaccines are rolled out and we can actually get serum from these test patients who have been injected with the vaccines, we can look at the comparability between the antibody response between naturally infected people and vaccinees. And I think that's going to be a really interesting study.
Thanks for bringing up the work of Dr. Ackerman and Lee. Are there other, can you comment on the overall response of the Dartmouth research community? We talked about your work and that work. and the work going on at DH with Dale. What about others at Dartmouth? How are they responding to this?
The response has been amazing, and it's what you would expect from this little community, which views itself as sort of the base camp to the world. And the incredible amount of enthusiasm and ingenuity that's come out from all corners of the school, in epidemiology, in Thayer School of Engineering, and within microbiology and infectious diseases, where you would expect. It's actually a very exciting time. I've been curating a small database of researchers so that we can coordinate our efforts and not overlap, and actually, provide each other with a forum for discussion. So there's a lot going on here, and it's incredible, because given just a few months ago, of course, there was nothing, but it's become quite a well-developed community.
Thanks. So that's, yeah, I mean, I've seen that too, and it's really exciting and gratifying. Dale, a question came in about the children, with that syndrome that appears to be related to, or maybe due to the cytokine storm. Any thoughts of use of lens for that?
Yeah, absolutely. We are trying to position to get into that clinical setting. So there is a lot of preclinical work. So that means in animal models, showing that GM-CSF is very involved in Kawasaki's disease. So there's been some work published showing that if you neutralize GM-CSF, and this is a mouse model, that you can prevent some of the cardiac myositis that results from Kawasaki's disease. So it's certainly an area that we're interested in. I think one of the bars is it's children. They tend to, even though they get very sick and end up in the ICU, the large majority of those kids, do fine. They end up being okay. So it's a little bit of a strategy of where do you intervene in those children. I know they're getting IVIG as one of the therapies, and that's kind of a standard of care for Kawasaki's disease. With these kids, it looks like there's a higher percentage that have IVIG refractory disease, so they don't immediately respond to that. So maybe that's an area where we could intervene with lens in those IVIG refractory settings. But absolutely, I think it's an inflammatory syndrome. It's clearly GM-CSF is involved in that syndrome, so it makes a lot of sense for us to try to see what lens could do for those children.
Great. Well, we're approaching the top of the hour, so I just want to close the session here. Thank you so much, Dale and David, for sharing your insights with us. And thank you all for joining us today. I'm sorry we're not able to get to everyone's questions. We encourage you to visit the Geisel and the Magnuson and the Office of Entrepreneurship and Tech Transfer websites to learn more about biomedical research and entrepreneurship at Dartmouth. Lastly, please remember to complete the 2-minute survey that you'll be receiving at the conclusion of today's program. Your feedback will help us to enhance future programming. Please have a wonderful rest of your day. Be well, be safe, and look forward to meeting some of you soon. Thanks again.
Thank you.